with a brief introduction. So I'm really pleased to introduce Dr. Giles Yeo today, who's visiting us from the University of Cambridge Metabolic Research Laboratories and MRC Metabolic Disease Unit. Dr. Yeo's scientific training began at UC Berkeley, where he got his bachelor's degree in molecular and cell biology with an emphasis in genetics. He then went on to do his PhD in the Department of Medicine at the University of Cambridge, where he worked with the Nobel Prize winner, Dr. Sidney Brenner, studying pufferfish genetics. He then did postdoctoral training also at the University of Cambridge with Susan Kenrick and Stephen O'Rahilly, where he really got into the genetics of obesity and notably made really important discoveries about mutations in the melanocortin 4 receptor gene, or the MC4R gene, that's implicated in obesity. Currently, he is a principal research associate and scientific director of genomics and transcriptional core at the University of Cambridge, where in his own group, he continues to make very important discoveries on the genetics of obesity, not only in humans, but also in other creatures, such as the popular Labrador retriever. And based on the title of your talk, I have a feeling we might hear a little bit more about that later today. And so before I turn it over to Dr. Yeo, I just wanted to say a few quick things about him beyond his science. The first is that Dr. Yeo is a truly gifted science communicator. And he does this with he communicates his science and the science of others to the general public through TV appearances, such as on the BBC, but also in writing through articles and a book, one of my favorite science books called Gene Eating. The second is that he's extremely generous with his time in this regard. So I used this book, for example, in a class last semester, and he spent time with some of my freshmen at the University of Pennsylvania, talking with them about his experience in science and in communicating science to the general public. And it was just such a treat for me and for the class. And third, like many others who study feeding behavior, Dr. Yeo loves food and loves cooking. And you can follow this on his Twitter, where he shares his really impressive homemade meals. And this is how I know that last night he had roast pork belly with an apple brandy sauce for dinner. And I'll just leave it at that. So go check it out. It's extremely impressive. And with that, I'll turn it over to you, Dr. Yeo. Thank you so much for spending our afternoon and your evening here at the Monell Chemical Senses Center. We're really looking forward to your talk.
Thank you so much, Amber. All right, so I was just telling Amber, I don't know if you heard in the let me just share this, where I had a nightmare 2 weeks ago. So I can see now people here on hopefully people can see my slides. Okay, so and let me just put on the show, and this is fine. And then what I tend to do is I then now close this little window here on the right hand side, so so I can get the talk. Anyway, so 2 weeks ago I was doing this in such like this. I think I was in Nice at the time. Just want to share where I am. I'm here in my office. That's my cowboy hat. But there we go. This is where I am. And I shut this thing off. And just as I shut it off, as I'm doing now, I dropped off the call. But I didn't know this, right? And so I gave a whole half-hour talk to nobody, like to the interweb. And I ignored my phone because I was trying to be professional. And anyway, I had to go back in and give another 15 minutes. Very stressful for everyone involved after I was done. I'm going to start now. I will stop weebling on. I'm shutting. Amber, you will shout if I have dropped off or I have not dropped off. I presume you can see me. There we go. All right, let's do this. So thank you so much, Amber, for inviting me. My name is Giles Yeo, and I'm based at the University of Cambridge. And I think the title is slightly cryptic. The brain control of food intake is not cryptic, but can an old dog teach us new tricks? So there are going to be two dogs which appear in today's narrative. There's an actual dog, and that's going to be obviously clear when it shows up. But there is also a metaphorical dog, and I'll highlight that as we actually go through this. And I'm a geneticist by trade, and I think being a geneticist is a perfectly upstanding thing to do. You know, my mother-in-law still speaks to me, so that's a positive thing. But when people ask what disease I study, and I tell them body weight, of which obesity sits on one end of the spectrum, immediately I become the bad person. I become the bad person because I'm perceived as giving fat people, overweight people, people living with obesity, terms I do not use in any pejorative fashion whatsoever, and excuse. And I'm just going to check that everyone is still there, which they are. Okay. And I'm now got PTSD by this, by the way. And excuse, which I think is always a very interesting thing philosophically, because if I was studying the genetics of dementia, the genetics of cancer, the genetics of arthritis, would I suddenly be giving those people an excuse? No. I mean, I'd be trying to understand biology, molecular mechanisms. God forbid, I might be trying to help somebody, right? But yet, when we talk about body weight, okay, suddenly I'm giving people an excuse. And that's because we think that body weight is a choice, okay? And I want you to take the next 45 minutes. I'm going to speak for 40 minutes, sort of like as a rebuttal of sorts as I go through some of the work that we here in Cambridge have been doing. And the other thing, which is of relevance here, is we now know, we within the field now know that by the very definition of studying the genetics of body weight is that we now know that we're studying the genetics of how our brain influences our feeding behavior. Okay, and I'll also bring in the evidence for that. Hence the title of today's talk. So my group is I'm an accidental neuroscientist, but my group is interested in how the brain controls food intake. And your brain needs to know, very simplistically, your brain needs to know two pieces of information in order to influence your food intake. It needs to know how much fat you have, because how much fat you have is how long you would last in the wild without any food. Okay, and these are going to be in the form of circulating adipokines, adipocyte hormones, and leptin being amongst one of them, which then signal to the brain. The brain then needs to know the second piece of information, which is how much you have eaten recently and how much you are currently eating. So these are going to be your short-term signals, and these signals are going to come from your gastrointestinal tract. Okay. Now these hormonal signals, or actually the nutrients themselves, circulate within the blood. They signal to the brain, which then responds and translates these signals, thereby influencing your next interaction with a menu, with a restaurant, or a fridge. Okay, and there are genetic modifiers that run throughout this process. And from there, we are then able to harness these genetic modifiers to try and understand biology. And I want to focus on one particular pathway today, which is the melanocortin signaling pathway. And this is one of the key fat sensing pathways that I was actually telling you about. So what is this melanocortin pathway? It is the leptin melanocortin pathway. They give it its full name. And it's when leptin is, which is an adipocyte hormone, which is secreted from fat in proportion to the amount of fat that you actually have. Leptin then signals to the brain, in particular a part of the brain called the hypothalamic region of the brain. Within the arcuate nucleus of the hypothalamus, leptin signals to the leptin receptor, influencing the processing of pro-opiomelanocortin by a number of prohormone convertases. These convert POMC into melanocortin peptides, which signal to the melanocortin-4 receptor or MC4 receptor, thereby influencing food intake and body weight. And I'll come back and give examples of all of that in a second. And how do we know that the melanocortin pathway is a key fat sensing pathway certainly within mammals, but also in other vertebrates? Because genetic disruption at every single level of this pathway, results in severe obesity, whether or not you have whiskers and a tail, or whether or not you don't have whiskers and a tail. And so I want to go and actually just give you a few examples of these genetic disruptions. But, and this is where the first old dog comes in, all right? This pathway, this old dog, is an old dog, because the vast majority of these genetic disruptions were identified before the turn of the millennium, before 2000, in a golden period of time between 1998 and the year 2000. This genetic disruptions of the vast majority of these genes here were identified. So hence the title of my talk. So, okay, well, this is so important, but we knew this before 2000. Can this old dog teach us any new tricks? And so while I'm going to give you today just examples throughout this process of the old dog, I also want to show you examples of some of the new tricks that they have revealed to us, and why why I still study the melanocortin pathway all these years on, because they continue to reveal to us new biology, additional nuance as well to the biology. So just so that everyone is on the same page, I've just introduced the melanocortin pathway to you. All right. So the melanocortin pathway, the crux of the melanocortin pathway lies with this particular hormone here, pro-opiomelanocortin. Oh, it's not a hormone, but it's a complex pro-polypeptide, and pro-opiomelanocortin, like many complex pro-polypeptides, is post-translationally modified into a number of different, post-translationally processed into a number of different biologically active moieties. And the melanocortin peptide elements of these are adrenocorticotrophin, or ACTH, and the alpha, beta, and gamma melanocyte stimulating hormone, or MSH. So there are 4 melanocortin peptides. And they mediate really quite a dizzying array, array of different biological effects via 5 different melanocortin receptors. Okay, so the MC1, MC2, 3, 4, and 5 receptors. Now you'll note that aside from the MC2 receptor, which is present on adrenal glands and is responsible for steroidogenesis and is monogamous for adrenocorticotrophin, or ACTH, all of the other receptors, MC1, 3, 4, and 5, are promiscuous for the various different melanocortin peptides that are there. Now, within the brain, which is where we're going to be focusing on, I'm going to be focusing today on the melanocortin 4 receptor. Not only are there endogenous agonists for the receptor, so the MC4 receptor, for example, beta, alpha, and gamma MSH, there's also an endogenous antagonist, AGRP, or agouti-related peptide. And it's a naturally occurring set of neurons that produce this antagonist, and it acts as a push-pull at the MC4 receptor in order to actually mediate energy homeostasis. And we know, okay, that POMC is important directly because human mutations, and this was identified, this child human mutations in POMC were originally identified by the group of Annette Grüters and Heiko Kruder from the Charité Hospital in Berlin in 1998, results in severe obesity. Actually, it results in really quite a complex phenotype because of the roles of the different melanocortin receptors. So the first thing you'll notice is bright red hair on this kid over here. Because of a lack of melanocortin action at the MC1 receptor. So the MC1 receptor is crucial for melanogenesis, okay? For that. The child has isolated ACTH deficiency because he lacks ACTH and actually has no capacity to produce quite a few of the steroids, and so has to be on steroid injections for the rest of his life, okay, because we need steroids. And this t-shirt is hiding a multitude of sins, but this child is also severely obese because of a lack of melanocortin action at the MC4 receptor. Now, I could show you now really a bit of work we did on the POMC knockout mice, which we created in the early 2000s, and they also recapitulated quite a bit of almost all of the phenotype that we actually see here. But I thought instead I would share. There we go. And this is an actual old dog, not that old of a dog, on our work and actually on Labrador retrievers. Now, this work was led by a veterinary surgeon colleague of mine, Eleanor Raffin, who did a PhD in human genetics, but then went back to becoming a vet. Now, Labrador retrievers are the most popular pet dog in the UK and in North America. Within the UK and of itself, 150,000 new Labrador puppies are registered every single year. Okay, in the UK, we use them to sell toilet paper. Okay, the Andrex puppy is a Labrador. But for any of you out here who actually own Labradors, you have to keep your compost bin shut because otherwise the Labrador will be in and with a shot and eats everything in sight until it nearly physically pops. Labradors are known to be tremendously food motivated. Eleanor, my veterinary surgeon colleague, was interested in what was driving this food motivation in these dogs. But to cut a long story short, we did throw the genetic book at this. Actually, a percentage of Labrador retrievers have a deletion in POMC. Now, it's not a complete deletion. So alpha, gamma, and ACTH are all still intact, and there's no adrenal problems. But it is a beta MSH specific deletion in effect. Okay, beta MSH and beta endorphin. But not all Labradors have it, and so we can do a genotype phenotype correlation. And what you can see here is these are wild type Labradors, heterozygous for the deletion, homozygous for the deletion, and this is body weight. And what you can see is that each of the deletion alleles is worth about 2 kilograms of weight, so that the homozygous, the Labradors homozygous for the deletion are on average about 4 kilos heavier than the wild type Labradors. Doesn't sound like a lot of weight, 4 kilos times nearly about 10 or 11 pounds, but you have to remember that Labradors only get to about 30 kilos. So 4 kilos is a lot of dog. And when we actually do the same thing with food motivation, okay, you see exactly the same pattern, clearly therefore driving the body weight phenotype that is there. And here is what I said. This is the effect size, a couple of kilos per deletion allele, or about 0.3 of a standard deviation of body weight. For those of you who follow the common obesity genetics, this kind of effect size is equivalent to 3 times the effect of the FTO risk allele. But as I said, not all Labradors have it. Okay, so certainly this is going to be true for UK pet Labradors. Roughly 20% are heterozygous, 2 to 3% are homozygous, with 75, 3/4 of Labradors, pet Labradors, being wild type. But the interesting thing is that 95% of Labradors are tremendously food motivated. And so now Eleanor is currently funded to throw the genetic book at this, whole genome sequencing, et cetera, in order to try and find other key genetic drivers. But undoubtedly, this deletion in POMC is a key genetic driver causing certainly a percentage of Labradors to become really quite obese, or at least increase their susceptibility for obesity. But so far, so not so interesting, really, because I've shown you that in 1998, we knew that in humans, that POMC was important for feeding behavior and therefore resulting in severe obesity with the mutation. We knocked the gene out in mice in the early 2000s and published the paper in 2003. So why am I telling you all of this? Well, there is actually a denouement to this story, which provides sort of like an interesting tale. Because not only are Labradors wonderful family pets with a fabulous temperament, which is why they're so popular as family pets, but they're also tremendously trainable. They are very, very trainable. And so much so that in North America and in the UK, they're primarily used as guide dogs for the blind. Okay. Now, guide dogs are like the Navy SEALs of the dog world. Okay, I mean they're trained within an inch of their life because they're about to be given a human being to look after for the rest of its life, and it's tremendous failure. Okay, so they're trained in the UK by a charity called Guide Dogs UK, and there's huge failure rates. And the ones that fail, the dogs that fail, become pets. Okay, so the few, the proud, the guide dogs, and they're trained with food using standard Pavlovian training techniques. Okay, and so given this heavy selection using food, 80% of successful guide dogs, certainly in the UK, have this deletion in POMC. So here's our concept, all right? So here's a guide dog bringing back visually impaired Mr. Smith, say. Imagine if a chicken ran across the road, okay? So then, well, what are the chances of a chicken dinner? 50-50, 80-20, doesn't matter, because the dog is trained, the successful guide dog is trained that it will have a 100% chance of dinner if he brings Mr. Smith home safely. So we started by looking at food motivation, which is obviously still what I'm interested in studying, but because of the selection pressure applied on the dog or the guide dogs, okay, highly selected for food motivated behavior, and suddenly we found ourselves in the realm of trainability. And so we ended up on the cover of Cell Metabolism. This is a chocolate Labrador, Jasper, one of our participants, and that is Eleanor who led the study. So when those of you who own Labradors, go home and you see Fido with his big googly eyes saying, Fido loves me. Fido doesn't love you. Fido is hungry. Oh, it's terrible thing to say. Anyway, so now you won't be remotely surprised if I tell you that actually mutations within the melanocortin 4 receptor result in severe obesity. And this is actually I joined Steve O'Ratley's lab in 1998, and this was my very first publication with him. And I ended up still being within the field. Now, okay, this is still an old dog. This is 1998. So has it taught us anything new? And actually, it has. All right. So we now know that mutations within the MC4 receptor form the commonest monogenic cause, or is a the commonest monogenic driver of obesity that we actually know about. All right. So we did a with so many of these mutations about. I'll tell you how common in a second. We now know that it influences not only it influences feeding behavior, and so this is work that we actually published together. Sadaf and I, Sadaf Faruqi, okay, and I published this work together in 2003, in which we were actually assaying the genotype phenotype correlation of mutations within the melanocortin 4 receptor and the resulting phenotype in human beings. And just to be clear, okay, so this was when I was still a postdoc. I did the work on the left-hand side in recombinant melanocortin 4 receptors within a lab in tissue culture, whereas the clinical phenotyping of the patients was done by Professor Sadaf Faruqi and her team in a clinical research facility. Okay, now the MC4 receptor is a G protein coupled receptor. It's tethered to cyclic AMP, so you could add ligand and measure like cyclic AMP. We had an assay. All right. Now, unsurprisingly, you're going to have severe mutations in the actual protein, deletions, removal of cysteines, introduction of proteins, that kind of thing, results in a complete loss of function mutation in the MC4 receptor. Okay. Whereas if you have more subtle mutations, then you end up with partial loss of function mutations in the MC4 receptor, anywhere from, I don't know, 30 to 70% loss of function. All right. Now, but what was interesting was that while Sadaf was actually characterizing the kids coming in as they came in, she did not put them into these different pigeonholes until I supplied her with the recombinant tissue culture luciferase assay, which is what I did. Okay, and what you can see here is that the percentage of function in this really quite in vitro assay in a lab was able to predict how much a child was going to eat in an ad libitum buffet scenario. So what did this tell us? This told us that the melanocortin 4 receptor was not a binary system. It was not on off. It was tunable. It was a rheostat, okay, where not only the number of copies of mutation on the surface of the receptor was important, surface of the cell was important, but the quality, the degree to which the receptor was dysfunctional or not, influenced the food intake and gave you a spectrum. And so this is now how common is common. So this is work that was that we've done in collaboration with a group in the University of Bristol, okay, and just got accepted last month and is now in press, in which we studied the melanocortin 4 receptor in this particular cohort, the Avon longitudinal study of parents and children, okay, or LSPAC. Now, this is a birth cohort. So between 1990 and 1992, all of the pregnant women within the area of Bristol, which is in the county of Avon, were recruited. Okay, and these kids had then had a number of different a number of studies done to them, including genetics, and then were then serially followed over time. So a couple of things. First, we ended up screening the MC4 receptor in all of in the entire or most of our LSPAC, and we found and then characterized the different mutations that we found. All right, and what we found was that 1 in every 337 people that we found, 0.3% of this particular, and this is a population study, okay, population cohort, 0.3% of people were heterozygous for a loss of function in the MC4 receptor. Now, if you scale that up to the UK, and there's no reason to imagine why the genetics, why the people around Bristol are going to be genetically different to any other white Northern European Caucasian human being in the UK, or probably in Europe for that matter. But if we scale it up, then 200,000 people in the UK are gonna likely to have loss of function MC4 mutations. Okay, so now what does this loss of function, 1 copy loss of function, mean? At 18 years old, because you remember this is a longitudinal study, This is an average difference in weight between carriers and non-carriers of nearly 18 kilos, 17.7 kilos, an average difference in BMI of nearly 5 BMI points, all right, and an average difference in fat mass of nearly 15 kilos at 18 years old between carriers and non-carriers. Now, this is an average. Some gain a little bit of weight, some gain a hell of a lot more weight. Now, when we actually look, this is the birth cohort, and so therefore they're followed repeatedly. And since this is 1990s, they're now in their 30s. But you can now look actually at them over time, these mutation carriers. Now, what I've done and what we've done now is to actually separate out the in dark blue are the complete loss of function mutations, so no cyclic AMP whatsoever. Okay, the partial loss of functions, and this will then be obviously can go anywhere, as I said, from 30 to 17% function, and the carriers of no mutations. And if you plot their growth curves, what you see is exactly what you expect to see, but it's still really, really cool. So the dark blue shows these are the complete loss of function. If you compare that to the light blue carriers of no mutation, that is an average BMI difference of nearly 10 points at 18 years old. And you see an intermediate phenotype with the actual partial loss of function. They're all heterozygous. These are just the degree to which you actually have a functional impairment in the MC4 receptor. And we've just taken some snapshots, now just a fat mass, just to show you that the vast majority of, to show you the difference in fat mass at 10 years old, at 14 years old, and at 18 years old. And you see that this happens really quite early on, in life. And so really 1 in 337.3%, white Northern European Caucasians, okay, that's the caveat here, is actually quite common. Now, we know that MC4, loss of function MC4 mutations have been found in nearly every single ethnic group throughout the world that have looked. And so we do believe that actually loss of function MC4 receptor is certainly going to be the commonest cause to date that we know of really quite severe obesity. Remember, at 18 years old, a difference of nearly 18 kilos in weight times 2.2, we're looking at nearly 40 pounds, okay, of difference because of one gene. Now there is actually a prospect of a treatment for this, would you believe? And so in 2016, the group of, once again, of Annette Grutus, Heiko Kruder, led by Peter Kuhnen, together with the Rhythm Pharmaceuticals, trialed a drug called setmelanotide in order to see, which is a melanocortin 4 receptor, MC4 receptor agonist. And what you see here, okay, is data from that, and it worked. So you can see here, these are the first 2 kids with POMC deficiency. This is what it was tested against. And you can see that this patient 1 over here, this child at 18 years old, was nearly 160 kilograms. That's a lot of kilograms. Okay, that's 340, 350 pounds. And here's patient 2 is at about 145 kilograms. All right. Now, patient 1 was then treated for 40 weeks, pardon me, just over 40 weeks with setmelanotide, and lost 50 kilograms in weight, okay, way more than 120 pounds in weight. And patient 2 was treated for 12 weeks and lost 22 kilograms. This level of pharmaceutically assisted weight loss has not been seen since the original leptin deficient patients were treated with leptin deficiency, and has really energized the field of melanocortin biology. Now, setmelanotide is approved by the FDA for rare obesity syndromes, including POMC deficiency, leptin receptor deficiency, and prohormone convertase 1, which is crucial for the processing of POMC deficiency. And now I know that it's in clinical trials against the many people with MC4R deficiency before moving actually to the general population. More new tricks every day, ladies and gentlemen. And now that we see that so many human beings actually have this melanocortin 4 receptor, it's been maintained in the population, there clearly is some advantage, we would imagine, to actually having it. And if you actually begin to look around us, we begin to see evidence of evolution. It's not sentience, I understand. Mother Nature using the MC4 receptor as a strategy to tweak feeding behavior. So for example, if we take a look at agricultural animals, and such as this particular breed of pigs, this is just a pig off the internet, okay? But like breed of pigs that make good bacon, and they make good bacon for two different reasons. They have a really good rate of growth naturally, okay, and a good fat to lean mass ratio, okay, thus making good bacon. And they have, and the farmers have just selected for these particular traits. They just happen to have mutations within the MC4 receptor. Unless you think that this is a weird mammalian thing, think again. Let me introduce you to those of you who don't know of them, the blind Mexican cavefish. Okay, so not only are these fish blind, not only are these fish, they're blind. Not only are they blind, they're Mexican. So I guess there are two questions. Why are they blind and why are they Mexican? So that big asteroid that hit the Earth, that killed all the dinosaurs, whacked into what is now the Gulf of Mexico. Okay, and so when the asteroid actually hit, it then caused all these cracks in the crust and what have you of the Earth, causing these underwater caves to form. Okay, and so in the Gulf of Mexico, and so what happened is then a number of fish got stuck into these caves. Okay, and so what happens in underwater caves? Well, there's not a lot of light, so the eyes evolved away because they didn't need the light. Okay, that's why they're blind. Now, the other thing which happens in underwater caves is a rather lack of food, or at least very, very little food. So any fish that was even remotely blasé about the sort of like the algae that kind of were floating by, or the plankton that was floating by and didn't go snap it up, became X-fish. Okay, so you needed to be pretty food motivated to survive in that environment. All of the surviving Mexican cavefish that today are blind because of a lack of light and have mutations in the MC4 receptor because that is what was selected to keep them alive. So here's my question for you, ladies and gentlemen. All right, where's the choice? These are pigs that were selected by human beings for a given trait. Labradors were selected for a given trait, not for a specific genetic thing, for a given trait. These fish were naturally selected because of where they got themselves stuck for a given trait. Enough food motivation to keep themselves alive, and it just so happened to be mutations within the leptin melanocortin pathway. All right, where's the choice? There is no choice. Okay, it's because of a specific set of adaptations they had made that they actually were got to where they were today. Now, when we actually look at the brain, what more can we tell, I guess, about the brain and these particular sets of neurons? We are always sort of like in a danger of grouping these neurons as some kind of homogeneous lump of cells, like POMC neurons, AGRP neurons. I did just that. Okay, when I drew you that summary, I showed leptin signaling to leptin receptor expressing POMC neurons, et cetera, to melanocortin 4. But this is obviously not the case. There is obviously a lot of heterogeneity within within these cells. And so now that they're drugs actually targeting the system, we have now become really interested in understanding the heterogeneity of neurons expressing the components of the melanocortin pathway. Okay, we started back in 2017 with POMC neurons. Now, this is before the era of DropSeq. So we fact sorted GFP expressing POMC neurons and sequenced them individually. So we got a couple hundred neurons. Okay, but this was enough to give us some interesting bits of information. The first bit of information we got was that a quarter of POMC neurons expressed the antagonist AGRP, which we always thought to be entirely separate, to be an entirely separate set of neurons to POMC, and they do exist as separate neurons. But clearly, some of the neurons express both POMC and the antagonist, and this wasn't an error because they grouped together. And for all intents and purposes, they looked, smelt, and behaved like AGRP neurons. And when we actually looked at the AGRP neurons, these are called AGRP POMC neurons, AP neurons, or POMC neurons by themselves, and just asked about some of their characteristics. So we looked, for example, expression of leptin receptor. Well, then the interesting thing is that we found that while 60% of AGRP leptin receptor expressing neurons, while 60% of AGRP neurons express the leptin receptor, only 12% of POMC neurons express the leptin receptor. And so what that told us was that while leptin would be influencing the behavior of POMC, its post-translational modification, et cetera, it is unlikely to have happened directly on leptin receptor expressing POMC neurons, but probably via AGRP neurons expressing leptin receptor. And when we look at the insulin receptor, what we find, however, in contrast, is that both AGRP and POMC neurons express similar percentages. About 60% of these neurons will express the insulin receptor. And these are transcriptomic signatures, but when we looked at it from a protein level, we saw exactly the same numbers come up. Now, what can we do with such data? Well, we can then begin to ask questions about POMC neurons, for example, which we know to be targets of drugs. So for example, this is the 5-HT2C receptor, which is the receptor for a drug which used to be on the market, lorcaserin, but has now been removed. This is the GLP-1 receptor, and this is the receptor for other drugs that are available, such as lorcaserin and semaglutide. Not lorcaserin, sorry. Semaglutide and liraglutide, the GLP-1 receptor agonist. So you can ask, so hang on a second, we know that these two classes of drugs target the POMC neurons, for example, but do they target the same POMC neurons? And the answer is no. There's very, very little overlap between POMC neurons that express the GLP-1 receptor and POMC neurons that express the 5-HT2C receptors. So they're likely to be functioning via independent routes. So this is one example of what we can do with these data. Now, POMC neurons are not only expressed within the hypothalamus, they're also expressed within the hindbrain. And so in work that we just actually is in press press now, we did and I didn't present today, we did a survey, specifically the area postrema and the nucleus of the tractus solitarius, the NTS, using nuc-seq, single nucleus RNA sequencing. And we did a survey of 16,000 cells in the fed and fasted state in mice, okay? And in and amongst these 16,000 cells, we found a number of POMC neurons. So, but not only neurons, but also you can see that the neurons all sit in a group over here. This is hindbrain neurons now. Here are the oligodendrocytes, here astrocytes, microglia, epithelial cells. And you can see that there's POMC represented in all of these different cells within the mouse hindbrain. Now, when we just pull out all of these POMC cells and look at them, we can see here, group 3 over here, these are the POMC cells, I can tell you, from oligodendrocytes, whereas these are going to be the neuronal POMC cells, okay? So POMC neurons. And so are these actual POMC neurons? Will they produce melanocortin peptides? So what we did was we checked for the presence of these prohormone convertases that are so important for the conversion of POMC into melanocortin peptides. Here's PCSK1, here's PCSK2, and what you can see is that almost all of the POMC neurons express either one or both of PC1 or PC2, but none of the oligodendrocyte POMC neurons express PC1, and 4 of the oligodendrocyte POMC neurons express PC2, okay? And if we then look at, and we can then make comparisons between the hindbrain POMC neurons with the hypothalamic POMC neurons. And one thing does stand out. So if you actually look at 50% of hypothalamic POMC neurons express the MC3 receptor, for example, whereas none of the POMC neurons within the hindbrain express the MC3 receptor. And there's a whole cornucopia of other data present within that paper, but I thought I would highlight the melanocortin elements of that paper. So, and just as a quick sort of swing to the right, can we make fruit flies fat? So because we were also interested, given that we were now seeing lists of genes that were appearing either from genetic studies or from transcriptomic studies, we really needed an in vivo way of assaying of feeding behavior. And so I'm not a Drosophila biologist by any stretch of the imagination, but we ended up developing a composite screen for relatively high throughput screens. So, for example, we use this assay called the CAFE assay. And for those of you unfamiliar with the CAFE assay, that is where you put a capillary and fill it with yeast and glucose, which is a favorite food of flies. And if you then see, we have actually marked where the meniscus of this is going to be. And if we put a known number of flies in there, and as they actually drink the food, you can then see a feeding a rate of feeding. Okay. And so here, if we place mice on a normal flies on a normal diet and put them into the setup, this is how much they would normally and typically eat. Okay, X number of millimeters per fly per period of time. When we fast them overnight, they eat more. When we put them on a high fat diet, they eat less. So the assay works. Other assays include the green stomach assay, in which flies, as it turns out, have a translucent stomach, where we actually fast the flies, put them on normal food for 20 minutes, and then put them onto dyed food for an additional 15 to try and identify a response to fast. And we also obviously just measure mass. Okay. So what we did with this particular assay was we looked therefore at HGRP neurons and POMC neurons and identified genes which change reciprocally in response to a fast. Because remember, these neurons act in opposition to each other and so we were trying to identify transcripts that were reciprocally regulated, up in HRP neurons and down in POMC neurons and vice versa. And then we looked for, I won't bore you with the details. We then looked for some of these genes which exist within the flies, and then we knocked the genes out in the neurons, knocked the genes down in the neurons of these flies, and then subjected them to the various assays, and then sort of did a heat map and produce a composite score. And we were able to find a number of genes that even in flies that are reciprocally regulated within these neurons that when knocked down resulted in a demonstrable phenotype in feeding behavior and in mass. So in the last couple of minutes, look, the hypothalamic appetitive circuitry, which I've discussed with you today, including the leptin melanocortin pathway, has been primarily worked out in mice for obvious reasons, okay, for lack of available tissues. But mice are not humans, although we do know that the main players all do exist in humans. Obviously, from genetic perspective, from pharmacological perspective, we know that they exist in humans, but what we don't know is the exact circuitry and whether or not the neurons that express these receptors, how similar or how not similar they are between humans and mice. And so we have begun a program now of using NucSeq, the same technique we use to survey the hindbrain of the mouse, to do the same thing in the human hypothalamus. And so this is one of our earlier runs. We now have a lot more data, but this is just one of our earlier runs in which we looked at 1,500 human hypothalamic neurons and 1,300 non-neurons. And if you focus on the neurons, you can see that even looking at the nuclei, we're able to identify GABAergic and glutamatergic neurons. And actually, we now also know that at least in this particular individual, there are 10 times more insulin receptor expressing neurons than leptin receptor expressing neurons within this part of the hypothalamus, and we are collecting more data a lot more data as we speak. We're also using now RNA-scope to actually map, therefore, key players of the appetitive control pathway back onto intact human hypothalami. So here is something which we published together with Frank Ryman and Fiona Gribble, in which we were mapping GLP-1 receptor and GIP receptor within so this is just an example, within the human hypothalamus. And here you can tell, for example, that we've identified co-expression within the human hypothalamus of hypothalamic neurons expressing both the GLP-1 receptor and the GIP receptor. And we're now in the process of doing exactly the same thing for the hypothalamic pathway within the human hypothalamus. So, and in the last, just a couple of slides, this is not going to be a long polygenic talk. Don't worry about that. But all of the initial genetic perturbations I've talked to you about are monogenic causes of obesity. So in other words, you have a mutation and you have a phenotype and you have severe obesity. But clearly, aside from the 0.3% of the population we think carry mutations in the MC4 receptor, the vast majority of us are the body weight we are, not because of single gene defects, but because of very subtle changes in any number of different genes. And we now know of over 1,000 genes that are or 1,000 loci that are linked to body weight. So this and common and to link to a normal distribution of body weight. Now, this Jackson-Pollock diagram here, there's no need to look at the individual genes here. I just want you to take away two pieces of information from this diagram. First of all, that there are two groups of genes. There's a pink blob up here, and there's a blue blob down here. Now, as it turns out, the pink blob highlights genes which are associated with waist to hip ratio. Okay, circumference of your waist over the circumference of your hip. And if you take the ratio, it indicates body shape. Do I have a big bum? Do I have a big tummy? Do I look like a sausage? Okay, this is where you put your fat. And the big blue blob down here are genes which are linked directly to BMI. They're linked to BMI, okay? And these are genes which have to do with how much fat you have. So this is where you put your fat, your body shape, and this is how much fat you have. Two messages. First message, where you put your fat has little, if anything, to do with how much fat you have. That's the first piece of information, okay, at least genetically. And how much fat, where you put, message number two, where you put your fat has to do with genes that are expressed primarily within adipocytes, okay? So in other words, primarily where you put your fat has to do with fat biology. So far, so not so surprising. But how much fat you have, the genes which are linked to BMI, how much fat you have to do with genes primarily that are expressed within the old noggin, within the brain, and where we know their function influences feeding behavior, okay? Now, this is a gene, FTO, which I've worked on since 2007. This is the first time I've mentioned it because it's a very sad story. And this is an uplifting moment of sharing data. So I don't want to share sad stories with you other than to stress that these are polymorphisms. These are not mutations. So half of you here will have the risk variance for FTO, making you on average 1.5 kilos heavier than someone without the variant. 1/6 of you, or about 1 billion people in the world, will be homozygous for the FTO risk allele, making you on average 3 kilos heavier, or 50% more likely to become obese over time. These are not mutations. All of you will have some mix of all of these genes, reflecting where you sit on the normal distribution of body weight. I share all this with you only to show two in particular, okay? So these are genes that all of us will have some mix of. Here on the left-hand side, we see POMC of Labrador fame. And then over here, we will see the melanocortin 4 receptor, okay, of blind Mexican cavefish and pig and human beings fame. This is the leptin melanocortin pathway. So now we know that severe mutations within the leptin melanocortin pathway result in a severe phenotype, as I've shown you ad nauseum today. But very subtle changes within exactly the same pathway ends up influencing where you sit on the normal distribution of body weight, right? So I'm gonna skip all this. So am I giving anyone an excuse? Folks, you got to consider your genes like a hand of cards, all right? You can have good hands, you can have bad hands, and the only people you can blame are your folks because that's where the genes came from. But you can win, even though it's more difficult, with a bad hand of cards. Let me give you another analogy. I will never, ever run as fast as Usain Bolt, and it's because of my genes and I'm sticking to it. But it doesn't mean that if I train, I won't run faster than I do now. People misunderstand what we do with genetics. They think that your genes give you a point in space and time. They determine who you are. And that's just not true. Okay. Your genes clearly bracket a set of possibilities. Of course they do. Why am I bald? Because of my genes. Why do I look Chinese? Because of my genes. But within that bracket of possibilities, you can move up and you can move down for any number of different reasons. How rich or poor you are, your lifestyle, choices you may make, whatever that situation may be. So are we all sinners? This is my last thought before I shut the hell up. Because what is a sin? A sin is something bad which you do by choice. That's what a sin is. So this is me choosing to eat a pizza. So body weight is not a choice, and I keep saying this. And you might be pointing out it is a choice. I am choosing to eat the pizza. Look at that. That's a choice, right? But let me just give you this closing couple of thoughts. We do not gain or lose weight overnight. We just don't. Okay, any given meal is not going to change your weight. Our body weight is a function of thousands of different feeding decisions we have taken over the past few years. Okay. Now we know, because of our genetic hand of cards, that some people are slightly less likely to say no. They find it more difficult. They won't say no, not twice as much, maybe a few percentage points, like 5% of the time, they will find it more difficult to say no. 5% over thousands of different feeding decisions is hundreds of thousands of calories different in what somebody eats versus somebody else. That is why we are small, medium, and large in this food environment we live in. Over the scale of time that is required to change body weight, feeding behavior, ladies and gentlemen, is not a choice. This doesn't matter. This doesn't mean we can change physics. We can't change physics. Mrs. Smith with type 2 diabetes that needs to lose weight, otherwise she's going to become blind and get her feet chopped off, still needs to lose her weight. She still needs to eat less. There's no magic. But until we as society understand that for some people, it is always going to be more difficult to say no than others, that for some people, okay, who are living with obesity, they just happen to be, they're not bad, they're not slothful, they're not morally bereft, they just happen to be fighting their biology. Until we as society understand that, we will never, ever be able to fix the problem of obesity that we're in at the moment. This is me choosing to eat a broccoli. So, ladies and gentlemen, thank you so much for listening to me. I want to thank my colleagues, Stephen O'Ratley and Tony Cole, Alan Raffin, I've mentioned. The guys in bold are folks that have done the work. And you can see all of my, like a Formula One driver, all of my various funding bodies. It's been a pleasure to speak to you. I'm happy to do some questions. Hopefully I haven't gone on for too long. Thank you so much, guys.
Thank you so much for an incredible and very thought-provoking talk. We can open for questions now. Please either put your questions into the chat, or you can direct message me and say you want to come on and have a question, or just unmute and hop on and join the conversation. And I think I will take the host prerogative to start. And actually, your ending really set up my question quite well. So you gave lots of different compelling pieces of evidence about how mutations in the melanocortin system leads to changes in feeding behavior, obesity in humans and other species. And so my question is, it's actually kind of based off of something that my lab has started to focus on, which are interactions between the hypothalamus and the reward circuitry. And so my question is, how do these changes in, for example, MC4R or POMC genes lead to changes in the reward circuitry? Has anyone looked at how these mutations cause changes in dopamine signaling in response to food? Or I know you've looked behaviorally at motivation, at least in the dogs, but at the neural level, are there interactions here?
I think the answer is going to be yes. Clearly, it's difficult to do this in a human because you can only do this via brain imaging. Actually, the one thing I'm probably not clear on is how much MC4R is directly expressed in dopamine expressing neurons. Maybe you can give me that answer. But I find it very difficult to believe that there won't be an interaction between a key appetite controlling pathway and the reward pathway, because we know that one influences influences the other. We know, for example, that if you actually have mutations in the MC4 receptor, and this I didn't, and this is work done by Sadaf, is that you are more likely, okay, to be drawn towards fat, okay, high fat food. So she set up the study, for example, a chicken curry study. And the reason why it was a chicken curry, it was because it was easier than to kind of like homogenize fat into the curry while everything looking and tasting the same. Okay, this is why they use fat. And what she showed was that if you came in wild type normal human being like me, or if you're a carrier of a mutation, a MC4 mutation, and you small, medium, large Goldilocks selection of curry and fat, that the MC4 deficient human being will always end up picking, liking, and finishing the curry with more fat. Now, is this reward? Is this texture? Is this reward and texture? Is it because there's a drive for fat? We don't know any of these answers, to be fair. But I would be deeply surprised if there was no interaction between the melanocortin system and the reward pathway.
Yeah, I mean, my kind of personal bias is I agree completely. I think the question is it going to be a direct relationship, like, as you said, MC4R or other kinds of genes expressed directly in the dopamine system, versus an indirect effect of how these genes in the hypothalamus change reward signaling through network activity. So I think that'll be really interesting as that develops.
I mean, do you know the dopaminergic system? I mean, whether or not it I feel I should know. It's terrible. I feel ignorant, but expresses MC4 receptors. Do Do you know the answer to that?
So I don't know. I'm sure with lots of single cell sequencing data now, there's lots of data. What we do know, at least from work that's come out looking at AGRP neurons, and I think POMC neurons too, there's no direct projections between these arcuate neurons, AGRP and POMC, and dopamine neurons in either direction, at least in the adult mice. And so we think these interactions are happening at least through indirect projections. But the kind of network interactions have definitely yet to be determined. So I think it's kind of an emerging field, but one where there's lots of work to be done.
Okay. Someone has appeared on screen. John Bentley has appeared on screen.
I have. Thank you for the talk. I had a question. So you had showed data looking at a lot of sequencing of human hypothalamic neurons and receptors for a lot of the known drugs for obesity, right? The serotonin receptor, the GLP-1 receptor, the leptin receptor. But the melanocortin system has effects outside of the hypothalamus. And where do you think, do you think the hypothalamus is the key center to look at, or do you think that it's part of, these other centers, and how do we get to the phase in which we're looking at the whole brain and the network brain obesity?
I mean, this is the $64 million question. I mean, I think it's easier to do so in a mouse, but even then, there is a lot of brain in a mouse. As it turns out, because we've just started the human work just over 18 months or so. As it turns out, the human brain is pretty large. That's the first thing, and it's very different from the mouse hypothalamus, and very different from the mouse brain. So I think we started with the hypothalamus purely because of real estate. It was just so much real estate there in order to, we figure, well, we may as well start there. We got another, just got funded to do a project now within the hindbrain. Once again, the hindbrain's a big area, but to look probably at the same way we did in the mouse, to look at the AP and to look at the NTS as well. I would love to look at the reward pathway, but that is a more diffuse, to my view, is a more a diffuse thing. But I think we're going to have to work our way in. Undoubtedly, it's going to be present in many other areas of the brain, and so we are. I don't want to sound like I'm the guy looking for my keys underneath the lamppost. That famous analogy, which is what we're doing at the moment. Oh, the hypothalamus is important, so we're looking there. So I agree with you. I think we do need to be looking at other parts of the brains, other parts of the brain as well, both in humans and in mice.
Okay, John, I have questions. Very nice presentation.
Thanks.
So in the last part, in the last part of your presentation, you mentioned your flat screen. I think it's very interesting. You use Elaviga, for your remark correctly, right, to knock down many genes, many hallmark genes from mice to identify.
Yeah.
And so, did you really see the flat obese phenotype? From your top list, as you put a fly in the high-fat diet, they definitely, based on screen result, they have increased intake of the high-fat, right? Did you see morphologically the fly, they look bulky?
No. So we certainly couldn't tell. It was not like the flies suddenly became round. No. So we did have to. We saw a difference in mass. We did measure triglyceride levels. So we did mush up the flies and measure triglycerides. There were difference in triglyceride levels. So no, they were not visibly. Larger, but they certainly weighed more, and they certainly had more triglycerides in them when we mushed them up and actually measured their triglycerides.
Okay, cool. So you use the Elaviga for the paneuronally knock down those genes. What about the fat specific, fat body specific GAL4 driver?
So we did not use the fat specific GAL4 drive for this, for a very reason that our understanding was, what the biology is telling us is that because BMI is largely controlled by the brain, because of food intake, we decided then to focus on genes that were expressed within the brains of flies. For that very reason, I think undoubtedly we will find different phenotypes, particularly if we were working on some of the adipocyte genes, for example. Then I think we would remove them from the fat body of the fly. But because we were working on BMI genes largely, and genes that were expressed, within the neuronal context, this is why we stuck with knocking the genes down specifically within the brain of the fly.
Okay, cool. Also, another really question is the interesting receptor MC4, right?
Mm.
Yeah, you study
The fly doesn't have an MC4R, just in case you were asking.
No, just religion.
Right.
So you focus on the brain. What about the MC4R in other tissue, let's see, white or brown fat or liver?
So
Is
the
it
MC4 receptor is a central nervous system specific. There's some arguments whether or not it's expressed in some gut cells, maybe, perhaps, kind of, sorta, but primarily its vast majority of expression is CNS.
Okay.
Yeah.
Okay, that's fine. Thank you.
Thank you.
And we are just about out of time, but I wanted to follow up with one last question that it's kind of comes off of Nick's question about the site of action for the MC4R in human obesity. And I was really struck by your single cell sequencing in human tissue. I think that's a really exciting future direction. So what kind of spatial resolution can you get with this this single cell or single nucleus sequencing in the human tissue, and can you use that to kind of get at where is it hypothalamic versus hindbrain the mutations are that are leading to body weight phenotypes in humans?
So what we're doing, once again, the human hypothalamus is a pretty big space, as we now find out. So what we're doing at the moment is not just taking out the whole chunk of hypothalamus. We're doing actually, so I don't do this work. We have a human pathologist, okay, that has to do it because he can get access to the brains, and he actually uses a dental drill and go and drills out the bits. And so what we've done is we've sectioned out the hypothalamus in 3 bits. Okay, there's obviously more bits, but this is the best we can do. Medial basal, which includes ARC and probably some VMN as well. Okay, where the PVN is, and the lateral part of the hypothalamus. We're at the moment double sampling the medial basal, once again, because we like that region and we think there's going to be a lot of mileage to actually identifying, and then sequencing the PVN region and the lateral region. So we will have region specific, both neurons and non-neurons, I want to point out, within those areas. We will then apply the same thing to the hindbrain. We haven't started the hindbrain project yet. And eventually, maybe if we get the funding to go to reward pathways, because that's the holy trinity, right? The hindbrain, hypothalamus, and the reward pathway eventually, and then we'll see what we see.
But you can get kind of subregions of the hypothalamus and have some kind of spatial resolution with
a pathologist involved. Trust me, it looked like a big mush to me. I can't say, and it doesn't look anything like that. I can tell you for free, the architecture does not look anything like a mouse architecture.
Well, that'll be really really interesting to see that develop. Thank you so much again for spending